Recombinant DNA technology producing DNA fragments. The most significant scientific advance in recent years has been the development of recombinant DNA technology that allows genes to be manipulated, altered, and transferred from organism to organism, even to transform DNA itself. These techniques have enabled us to understand better how organisms work and to design new industrial processes and medical applications. A number of human diseases result from individuals being unable to produce themselves various metabolic chemicals. Many of these chemicals are proteins, for example insulin. They are therefore the product of a specific length of DNA, that is, the product of a gene. Treatment of such deficiencies previously involved extracting the chemical from a human or animal donor and introducing it into the patient. This presents problems such as rejection by the immune system and risk of infection, as well as high costs. It follows that there are advantages in producing large quantities of pure proteins from other sources. As a result, techniques have been developed to isolate genes, clone them, and transfer them into microbes. These microorganisms are then grown to provide a factory for the continuous production of a desired protein. The DNA of two different organisms that have been combined in this way is called recombinant DNA. The resulting organism is known as a transgenic or genetically modified organism, GMO. How then is it possible that the DNA of one organism is not only accepted by a different species but also functions normally when transferred? The answer lies in the fact that the genetic code is the same in all living organisms. It is universal and can be used by all living organisms. This explains why the coded information on the transferred DNA can be interpreted. But what about making of proteins? Well, that's universal too, because transcription and translation are essentially the same in all living things. As a result, transferred DNA can be transcribed and translated within the cells of transgenic organisms, and the proteins it codes for can be manufactured in the same way they would within the donor organism. This is all indirect evidence for evolution. The process of making protein using DNA technology of gene transfer and cloning involves a number of stages. 1. Isolation of the DNA fragments that have the gene for the desired protein. 2. Insertion of the DNA fragment into a vector. 3. Transformation, that is the transfer of DNA into suitable host cells. 4. Identification of the host cells that have successfully taken up the gene by the use of gene markers. 5. Growth and cloning of the population of host cells. Before a gene can be transplanted, it must be identified and isolated from the rest of the DNA. Given that the required gene may consist of a sequence of a few hundred bases amongst the many millions in human DNA, it's no mean feat. There are several methods of producing DNA. 1. Conversion of mRNA to cDNA using reverse transcriptase. 2. Using restriction endonucleases to cut fragments containing the desired gene from the DNA. And 3. Creating the gene in a gene machine, usually based on a known protein structure. Using reverse transcriptase. Retroviruses are a group of viruses of which the best known is HIV. The coded genetic information of retroviruses is in the form of RNA. However, in a host cell, they are able to synthesize DNA from their RNA using reverse transcriptase. It is so named because it catalyzes the production of DNA from RNA, which is the reverse of RNA into DNA into RNA during transcription. The process of using reverse transcriptase to isolate a gene is straightforward and requires RNA, reverse transcriptase, DNA polymerase, and finally you end up with a double-stranded piece of DNA. A cell that readily produces the protein is selected. In the case of insulin, you would choose beta islets of Langerhans cells. These cells have lots of mRNA. Then reverse transcriptase makes the DNA from the RNA, and this is called complementary or cDNA. To make the other strand of DNA, the enzyme DNA polymerase is added to make a complementary strand to give you double-stranded DNA. Using restriction endonucleases. All organisms use defensive methods against pathogens. Bacteria are frequently infected by viruses that inject their DNA into them in order to take over the cell. Some bacteria defend themselves by producing enzymes that cut up the viral DNA. These are called restriction endonucleases. Each type of restriction endonuclease will cut at a different sequence. For example, GTTAAC. If they cut and leave straight edges, it's known as blunt ends. Other restriction endonucleases cut DNA in a staggered fashion, and this is called producing sticky ends because there are unpaired bases. An example would be a recognition of AAGCTT. If you read the bases in the opposite directions, they read exactly the same, and we call this a palindrome. So if you're reading it from left to right, it reads exactly the same as if you're reading it from right to left. This feature is typical of the way that restriction endonucleases will cut the DNA to leave sticky ends. And finally, the gene machine. You can make a gene by knowing the desired sequence of nucleotide bases of a gene, usually from the desired protein, using a best guess. So if you know the sequence of bases needed for each amino acid, you first identify the amino acids in the proteins, then use the best guess to figure out what the DNA triplets would be. The desired sequence of nucleotide bases for the gene is then fed into a computer. The sequence is checked for biosafety and biosecurity to ensure it meets international standards, as well as various ethical requirements. The computer designs a series of small, overlapping single strands of nucleotides. These are called oligonucleotides, which can be assembled into the desired gene. In the automated process, each of the oligonucleotides is assembled by adding one nucleotide at a time in the required sequence. The oligonucleotides are then joined together to make a gene. This gene doesn't have introns or other non-coding DNA. The gene is replicated using the polymerase chain reaction. The polymerase chain reaction also constructs the complementary strand of nucleotides to make the required double-strand gene. It then multiplies this gene many times to get lots of copies. Then, using sticky ends, the gene can be inserted into a bacterial plasmid. This acts as a vector to be transferred into the organism. The genes are checked using standard sequencing techniques, and those with errors are rejected. The advantage of this process are that any sequence of nucleotides can be produced in a very short time with great accuracy, and these artificial genes do not have introns or other non-coding DNA which would not be able to be transcribed or translated by prokaryotic cells.